This lecture is on viruses and prions. So it's important to realize that viruses are non-living and they are infectious agents. So infectious means that a virus can transmit a disease between two individuals. Disease can be passed from one to the other, such as through a cough or a sneeze, or through body fluids. And non-living means that various characteristics of non-living, usually they do not have their own energy source, and so they are depending on a host, and they use multiple enzymes from the host. And then, so the definition of a host then is the body that the virus inhabits. So the living thing that the virus depends on would be called the host. Virology is the study of viruses. To appreciate this field, it is important to see it in a historical context. In the early 1900s, not a lot was understood about the structure of nucleic acid and protein, and it was still unclear which of the two were the hereditary material, really until the early 1930s. One of the early pioneers in virology and also in molecular biology is Wendell Stanley, who in 1935 made crystals of tobacco mosaic virus. And people were already making crystals of proteins at that time. And you can see the crystals in yellow on the slide. But an entire virus had not been crystallized. This virus is known to infect tobacco, which you can see in the little inset there. And in 1946, Wendell Stanley was awarded the Nobel Prize. The significance of his work was that it showed molecular biologists that not just living things could cause disease. And so obviously what he found was that the virus contained protein and nucleic acid, but at the same time was also capable of being infectious. And it also showed us that not just living organisms can divide and replicate. And so he had crystallized a pure nucleic acid protein complex called a nucleoprotein. And interestingly, even his pure crystals were capable of replication. But let's take a look at the structure and classification of viruses, or the grouping of viruses. All viruses have two essential features. One is the nucleic acid, which can be DNA or RNA. And of course, this is now known as the hereditary material. And all viruses are also surrounded by a protein coat, otherwise known as a capsid. And then viruses also may have another feature, which is called the envelope. Not all viruses have the envelope. The envelope is a membrane, which is a phospholipid bilayer. And that phospholipid bilayer may or may not be associated with glycoproteins. So in the pictures we have here, drawing a naked virus, which is composed of an icosahedron, which is a geometric shape with 20 triangular faces, and this is made of protein, and the viral capsid forms this shape, and then it encloses a nucleic acid. And this icosahedron may or may not be surrounded by a membrane which contains glycoproteins, many of which might be involved in recognition of the host. That's called an enveloped virus. The naked example we have here is of adenovirus, and the enveloped virus is the herpes virus, which is a disease that is transmitted through body fluids. This slide is just showing you some alternative shapes of viruses. You have the icosahedron, the 20 triangular faces, and also these helical viruses. Okay, it kind of looks like a coiled spring, which is shaped because of its nucleic acid that is on the inside. Rabies, measles, and tobacco mosaic virus are examples of helical viruses. And then in this slide, we're looking at the relative sizes of viruses. And so we start off talking about a protein, okay, which is about roughly the size of 8 nanometers, or 10 to the minus 8th meters. And let's look at that by comparison to a virus, such as a flu virus, which is maybe only about 80 nanometers, or 10 times greater than the size of a protein. But a bacterial cell is about 1,000 nanometers, and or otherwise known as 1 micron. And then if you compare that to an animal cell may be 50,000 to 100,000 nanometers in size. And then let's take a look at viral replication, which is a little bit different depending on whether the virus is a DNA virus or an RNA virus. So in this slide, we're going to take a look at DNA viruses. And so DNA viruses have a molecule of DNA, and that DNA is going to be transcribed into messenger RNA, which is then used to make protein. That's one way. Or the DNA can integrate into the host genome and become a provirus. So here we have the host genome shown in red, and then the DNA of the virus integrates into the host DNA. And it may sit there for a while, or it may immediately be used to make messenger RNA, and then the messenger RNA takes over the host ribosomes and is then used to make protein. And then RNA viruses are a little bit different. These are viruses that have capsid, but they also contain the nucleic acid is RNA. And so these viruses can either use that RNA as messenger RNA, which then takes over the host ribosome and protein synthesis machinery and is used to make viral proteins, which are then packaged. Or the messenger RNA of the virus is first transcribed into DNA, okay, or reverse transcribed by an enzyme called reverse transcriptase. This enzyme is usually carried by the virus itself. This is the way that HIV replicates. and then the DNA may integrate into the host genome, and then at a certain later time be used to produce viral proteins. Some infectious agents are even smaller than viruses, and on this slide we're talking about viroids. Viroids are the smallest particles that are able to replicate. They consist of a single short strand of RNA with no capsid, but they infect plants, and they're able to disrupt plant cell metabolism and destroy crops. Many economically important crop plants have been destroyed by viroids. Another very tiny infectious agent is the prion. The prion is just simply made of protein, and that protein is in an abnormal shape, which causes it to form clumps. And those clumps are inside cells, and they therefore disrupt the metabolism of the cell. Prions, there's a picture of a prion there, a computer graphic of a crystallized prion. Prions can also be found outside cells and found in the brain. They're composed of about 250 amino acids, and they cause various diseases, such as bovine spongiform encephalopathy, otherwise known as mad cow disease. And they cause another disease in sheep, which is called scrapie. And scrapie is characterized by a degeneration of the nervous system, causing the animal to develop tremors. And then you have the Creutzfeldt-Jakob disease in humans that is also a brain disease. This is a fatal disorder, and it affects about 1 person in every 1 million people per year worldwide. In the United States, there are about 300 cases per year. Usually appears later in life and runs a rapid course, which, as I mentioned, is terminal. And that ends our lecture on viruses and prions.